The next organelle we're going to look at is the nucleus. The nucleus also is going to include the specialized areas of the nucleolus and the bounding membrane or nuclear envelope. So the nucleus contains something called nucleoplasm and chromatin. Chromatin is, of course, the genetic material. The nuclear membrane includes little areas where things can enter and exit the nuclear region called nuclear pores, and it contains a specialized area of RNA production called the nucleolus. The nuclear envelope is just like the plasma envelope, the plasma membrane, is made of lipid bilayers. However, it is a double membrane which is folded back upon itself in the same way that an envelope is folded back upon itself. And you can see the little red arrows that are showing you those bilayers. So at a lower magnification like this, you really only see one dark line for each membrane, whereas if you were at a very high magnification with good resolution, you might be able to see both of the bilayers. So to the far left and the far right over here, let's see if I can get here it is, the far left area here, as well as the far right, you can see examples of nuclear pores. And these are very specialized regions that have a number of proteins that we're not going to discuss in any detail here, that regulate the passage of raw materials and other signaling molecules into the nucleus, and also regulate traffic leaving the nucleus, and that would be, again, a lot of signaling molecules, and as well as all of the RNA that is produced, the mRNA, rRNA, and tRNA. So ribosomes are comprised of rRNA, and they are a non-membrane bound organelle, and they are comprised of two subunits, the large and small subunits, and you can see here this kind of snowman model. Where is my cursor? Here it is. Usually you find ribosomes bound to the endoplasmic reticulum, which is why it's called rough endoplasmic reticulum, or you can see them free in the cytoplasm in little chains called polysomes. These are the organelles of protein synthesis. That's their really only purpose. So this little diagrammatic, I'm not going to go through all of the steps in protein synthesis. That will be covered by other disciplines, and you probably have already seen it before in your prior studies. However, this is here really just to have me remind you to go through those steps of protein synthesis and recall all of those details, how the RNA binds to the ribosomes and the signal recognition particles that are involved, and the initial portions of the proteins and how it then docks onto the RER and so on. All right, so just go through all of those and remind yourself of that process. So the rough endoplasmic reticulum is a membrane-bound organelle. It is, in fact, all membrane, large membrane-bound sheets. It's a series of sheets enclosing a single intramembranous space called the lumen, or sometimes called a cisterna. Those are equivalent terms in this case. And it's defined by a surface coated with ribosomes. This is the site where protein synthesis, well, a great deal of protein synthesis takes place. And within the lumens of the RER, protein folding and glycosylation happens via specialized enzymes that reside in that RER lumen. So here is an image, an electron micrograph of a region of cytoplasm which is simply packed with RER, and the ribosomes are pointed out. They are studying the outer surface, in other words, the cytoplasmic surface of the RER, and the inner or lumen cisterna surface of the RER is marked by the blue asterisks. There's another kind of endoplasmic reticulum called smooth endoplasmic reticulum. And this is more of a branching tubular structure that contrasts with the sheet-like structure we saw in the rough endoplasmic reticulum. The amount of smooth ER is very variable depending on the metabolic activity of the particular cell you're looking at and depending on the tissue type that you're in. So what does smooth ER do? It has a number of important functions. It controls lipid synthesis, especially phospholipids and steroids, glyconeogenesis, particularly in the liver, detoxification, and as well as calcium ion metabolism. So what does it look like? Well, like I said, it's a series of kind of interconnecting tubes. You can see the model on the right kind of shows you that interconnection, whereas the electron micrograph on the left just shows you a single plane of section of all of those kind of disorganized looking tubular structures. So recall, this is smooth. It does not have ribosomes studying it as RER did. In fact, you can see some free ribosomes or polysomes nearby. I think they are marked with the 3 in this micrograph. The next organelle is another membrane-bound organelle called the Golgi apparatus. This is, in appearance, a stack of membrane-bound disc-shaped cisterna, and usually it's surrounded by quite a lot of vesicular traffic on both the trans and cis face, as well as transferring on the lateral face from one stack to the next. So what does it do? The Golgi apparatus functions in protein modification. It also sorts and targets, all right, by modifying the proteins by adding little post-translational modifications that target them to specific locations either within or without the cell. So it can target them to the plasma membrane for export, or it can target them to other organelles. And it also packages them within small vesicles. It's closely associated with something called the trans-Golgi network. The trans-Golgi network is a network of interconnected membrane-bound vesicles that are really kind of like the FedEx system. They deliver to various portions in the cytoplasm to all the different areas. So a Golgi apparatus, here you have actually a series of Golgi apparatus arcing through this area here. So you see the various stacks here and lots of little vesicles nearby around them. So there's 1, 2, 3, 4, maybe 5 different Golgi apparatus kind of in a row here. And you can see all of their little membranes for transferring and sorting and targeting to various portions of the cytoplasm. So we'll switch gears here and talk about this process called endocytosis rather than a specific organelle right off. So what is endocytosis? Endocytosis is the uptake of fluid and macromolecules, and there are 3 different ways that cells do this. The first is called pinocytosis. That is the non-specific ingestion of fluid and small molecules in small vesicles. So As you can see in the little diagram here, material is taken up in small vesicles right at the plasma membrane, which are then transported into the cell, and they merge with various other structures within the cell that we'll discuss in a few minutes, and are then utilized. The micrograph to the far right has little arrows showing small pinocytic vesicles right underneath the plasma membrane. And this is very commonly seen. This is actually in a capillary, so you're seeing things being transported across the cell via pinocytosis. The second process is phagocytosis. Phagocytosis is the ingestion of large particles or cellular debris or even microorganisms like bacteria or virions via something called pseudopodia. So pseudopodia are false feet, or kind of like arms of cytoplasm that the cell reaches out to engulf particles or large things like bacteria. This process is typical of very specialized cells, which are called appropriately phagocytes, and many of them are involved in your defense system against microorganisms, and we'll talk about them as we get to the next block. So here in the diagram to the right, you can see that process occurring, whether it be a bacterium or particulate matter. The cell reaches out the little pseudopodia and engulfs the particulate matter or microorganism and brings it into the cell, where it then can break it down using lysosomal enzymes, which we'll discuss in a few minutes. So receptor-mediated endocytosis is the third process that cells use to get material inside of the cytoplasm. And this allows entry of specific molecules using receptors for those specific molecules which reside in the plasma membrane, and clathrin-coated pits and vesicles that facilitate the bringing in of those materials. So looking at this diagram to the right, you can see how the receptors cluster together, and then due to the binding of clathrin on the inner surface, you get the formation of a coated pit, which then, the more clathrin and the more receptors are involved, can then invaginate into a vesicle and be brought inside the cell. Eventually, those clathrin molecules come off of the outside and are recycled back to the membrane, and the uncoated vesicle can then be fused at the appropriate place, usually with either a lysosome or what we call an endosome, which is a compartment that we'll talk about in a couple of minutes. So here is a really nice resolution set of images of coated pits and vesicles showing, you can see at the top here, the cargo or the substance that's being bound to its receptor, and then on the cytoplasmic surface you can see these kind of little spiky molecules here, which are the clathrin, which coats that, and then as it invaginates further and further, you form the vesicle, and then eventually that gets brought via this membrane fusion here, gets brought into the cell cytoplasm. And here at lower mag you can see several examples of clathrin-coated vesicles in these cell